Hi, I'm Timofey Prodanov, my co-author Vikas Banzal. We are from the University of California, San Diego, and we would like to present you a robust and accurate estimation of copy number for duplicated genes using whole genome sequencing. Human genome is highly repetitive. It contains long segmental duplications, and the duplications cover a large portion of human genome and around 1,000 protein coding genes. Segmental duplications are problematic for short read sequencing because read mappers don't have a good idea where to put reads, and this can lead to problems in downstream analysis such as copy number variation detection or single nucleotide variation detection. One of the duplicated genes is SMN12, and mutations or copy number changes in this gene can lead to spinal muscular atrophy. And paralog-specific copy number matters because if an individual has two copies of SMN1 and one copy of SMN2, it would be a healthy individual, and the other way around, it can be an affected individual. One of the useful definitions is paralogous sequence variants, or PSVs, and they are a small difference between repeat copies. The problem is that PSVs often coincide with polymorphisms and can be unreliable. Finally, we want to define what copy number is. In n-copy duplication, paralog-specific copy number, or PSCN, is a tuple of length n, and this stores copy number for each repeat copy. And aggregate copy number is a sum over paralog-specific copy number. Our tool, Parascopy, takes whole genome sequencing for multiple samples as input, and also a list of duplicated regions of interest. For each region, we calculate aggregate copy number and parallel-specific copy number profiles. Here you can see a short pipeline of the method. We use a pre-computed database to find all the repeat copies of a duplication and those PSV positions. And for each sample, we calculate background read depth using unique regions of the genome. Next, for a duplication, we remap reads from all the copies to the first copy, and this allows us to calculate read depths independently of read mapping and read mapping quality. And I will explain next steps in more details. So we start by calculating hidden Markov model to estimate aggregate copy number, and we use multiple samples to refine model parameters, for example, initial or transition probabilities. And in this example, 3 samples have a deletion in the end, but only 2 of them were identified on the first iteration. After we refine model parameters, we can identify deletion in all 3 samples. Next, after we know aggregate copy number, we can calculate parallel-specific copy number, and we use PSV allelic read depth to do that. The problem is that if we take all the PSVs and all the samples, they can be inconsistent with each other because there are unreliable PSVs and because there are samples that are non-reference copy number. Because of that, we use EM algorithm where parallel-specific copy number is a hidden variable and PSV F values are parameters, and F values are frequencies of the reference allele for each repeat copy. And what is important is that if PSV is reliable, then all its F values are close to 1. We can analyze different loci independently, and because of that, we can run Parascopy in parallel. It takes around 19 hours to calculate copy number profiles for 700 samples and 167 loci using 16 threads. Additionally, we can use pre-computed model parameters to analyze new samples. It takes around 25 minutes to analyze a single sample. Next, we wanted to validate Parascopy results using experimental data. On this slide, we can see comparison with MLPA aggregate copy number estimates for SMN12 genes. There are 2 observations for each sample because there is a common deletion. And in 972 samples that we looked at, there is 100% concordance between Parascopy and MLPA. On this slide, there is an extended table. There are 4 disease-associated genes and 3 methods. Parascopy, QuickMare2, which provides parallel-specific copy number for the whole genome, and a custom method. So these are methods that are designed specifically for one gene. And there are 3 different custom methods here. And in all cases, you can see that Parascopy provides either equal or higher accuracy compared to other sequencing-based methods. Next, we looked at reliable PSVs. So on this plot, you can see PSV is reliable if points on both subplots are high. And Parascopy detects 10 to 19 PSVs as reliable across 3 populations and 0 in African population. And in red, there are 8 PSVs that were reliable in SMN copy number caller. So this is a method designed specifically for SMN12. And we detect all of these PSVs as reliable, but we also detect some additional PSVs based on the population. Additionally, it's very important to see that there are more than 40 PSVs in this region, and if a method is not designed to detect reliable PSVs, it will make errors because more than half of the PSVs are unreliable. Another interesting case is AutoA. It's a disease-associated gene, and in the reference there are 2 repeat copies. However, you can see on this plot that the most common aggregate copy number is 6, which corresponds to 3 repeat copies. So this means that there is a missing copy in the reference. And additionally, we looked at a novel CHM13 assembly, and it has 3 repeat copies. In conclusion, parascopy uses multiple samples to estimate aggregate copy number. We find reliable PSVs and use them to estimate paralog-specific copy number. Parascopy has higher or equal accuracy compared to other methods, and parascopy can analyze large number of loci. And we are not designed specifically to analyze only one gene. You can find parascopy on this link, and full presentation will be on Tuesday at 11:40 UTC. Thank you.